I would like to present a research about effect of peptides from plasma of patients with coronary artery disease on the vascular endothelial cells. The research was done at Cornell Health, William Beaumont University Hospital, and Taras Shevchenko National University of Kyiv, Ukraine. The whole research team doesn't have any relevant financial disclosures. The objective of this study was to characterize the effects of peptides collected from the blood plasma of healthy subjects and patients with angina or myocardial infarction on endothelial cell secretion using in vitro model. As we know, the appearance of specific plasma peptides in coronary artery disease may induce the release of vasoactive molecules by endothelial cells. This can shift the homeostatic balance towards the procoagulant state. As part of the compensatory response under thrombogenic conditions, these peptides may affect endothelium to release profibrinolytic molecules to maintain a hemostatic balance. Background: endothelial cells are constantly exposed to the influence of substances present in blood. One hypothesis is that peptides, among other molecules present in the circulation, are more likely to affect endothelial function. It's known that the amount and repertoire of proteins in biological fluids change dynamically according to the physiological or pathological state of the individual. Some endogenous substances such as cytokines, modified proteins, antibodies can induce alterations in endothelial properties, indicating the potential relevance of those mechanisms to cardiovascular events. Methods: in our study, we included 70 patients hospitalized to the cardiology department of Kyiv City Hospital number 12 in Kyiv with the preliminary diagnosis of coronary artery disease. We were able to collect blood samples on the day of admission before beginning any treatment. Human endothelial progenitor cells were incubated for 24 hours with peptides isolated from the plasma of healthy subjects of patients with stable angina, progressive unstable angina, and myocardial infarction. The contents of some soluble anticoagulant and procoagulant mediators in human endothelial progenitor cells were treated with peptide pools and then compared. Results: peptides from the plasma of patients with myocardial infarction promote endothelial cells to release both von Willebrand factor and endothelin-1. However, peptides from the plasma of patients with unstable angina suppress the secretion of endothelin-1 and increase the secretion of von Willebrand factor and tissue plasminogen activator. Conclusion: comparison of the plasma proteins of patients with myocardial infarction or angina and healthy subjects is a promising strategy for detecting either coronary artery disease biomarkers or triggers of cardiovascular events.